Welcome to our quantum biology podcast. Our big question today: does quantum tunneling shape both cutting edge nanotechnology and certain processes in living organisms?
It's uncontested in nano-devices, but far from settled in biology. Some embrace the notion that tunneling speeds up enzymes or mutates DNA, while others dismiss these ideas as hype.
We'll explore the agreements, the controversies, and the reasons each side remains passionate. Along the way, we'll highlight short questions and answers, then transition to deeper discussions. Maya Hadid asks,
That's right. This is a topic that unites physicists, biologists, engineers, and chemists, and sometimes pits them against each other. Let's begin with nanotechnology, where quantum tunneling is firmly established.
in semiconductor manufacturing, we want transistors at ever smaller scales, but tunneling can be either helpful or disastrous. Any counterpoint?
Precisely. It's the backbone of advanced components like tunnel diodes, yet it can also ruin circuit efficiency if it's uncontrolled.
At the nanometer scale, classical physics predicts electrons shouldn't cross insulating barriers. However, quantum mechanics shows that electrons have a finite probability of appearing on the other side of these barriers, a phenomenon called tunneling. We harness this effect in devices like single-electron transistors, which can process signals with extreme precision. They manipulate electron flow at the level of individual particles, opening the door to novel computing architectures. At the same time, engineers struggle to minimize unintended tunneling, often leakage currents, that wastes power and generates heat. This battle becomes more intense as transistor gate length dip below 10 nanometers. The challenge isn't whether tunneling exists, it definitely does, but how to master it in a way that boosts performance rather than causing unwanted losses.
That sums it up nicely. Graphene and other two-dimensional materials are prime examples of new approaches. By structuring these materials cleverly, we can direct electrons in ways that harness tunneling constructively while limiting the downsides. Researchers are also experimenting with stacking different 2D layers to build van der Waals heterostructures, which in theory could finely tune tunneling probabilities. If these efforts succeed at scale, future computer chips might rely heavily on the quantum realm to maintain efficiency and speed.
And it's not just about computing. Quantum tunneling in nano-devices opens possibilities in sensing and energy conversion. Some concepts aim to use tunneling currents for ultra-sensitive detectors capable of identifying single molecules of gas or even single photons of light. That merges well with biological applications where detecting tiny amounts of biomarkers is crucial for early disease diagnosis. Though the solid foundation in nanotechnology might be a stepping stone to bridging quantum concepts with biological systems.
Which leads us to the question, is it feasible to transfer these concepts directly to complex, messy biological environments?
That's where the debate begins in earnest.
Let's shift gears. Is quantum tunneling truly significant in enzymes?
Some claim it explains the near impossible speed of certain reactions. Others argue that advanced classical theories are more than adequate.
Enzymes can improve reaction rates by factors of millions or billions. One proposed mechanism is that protons or electrons tunnel through energy barriers within the enzyme's active site rather than going over them. Proponents of this hypothesis often point to the drastic slowdown observed when hydrogen is replaced by deuterium. Classical physics can't always account for such a steep change in reaction rate. However, critics counter that the enzyme environment has elaborate electrostatic landscapes and dynamic conformational changes. These can mimic quantum-like effects without requiring direct tunneling. Moreover, any quantum state formed in a biological system must contend with continuous thermal noise, chaotic solvent interactions, and the flurry of other molecules inside a cell. That environment is incredibly rough on delicate quantum phenomena.
Indeed, the central question is whether the fleeting coherence needed for tunneling can survive under physiological conditions. Biologists skeptical of the tunneling explanation say, look at how well classical catalytic theories have served us. They predict rates accurately across many enzyme systems. We only see these anomalies in a handful of carefully designed experiments, often in artificial or simplified conditions. On the other hand, quantum advocates argue we might be underestimating the subtle microenvironments that enzymes can generate, which might preserve tunneling briefly enough to affect reaction kinetics.
Exactly. Both sides present valid points. It's also worth noting the difference between quantum mechanical tunneling in a strict sense and the broader notion of quantum effects. Sometimes the term quantum is used loosely in discussions of enzyme catalysis. That muddles the debate because it's not always clear if people are talking about genuine tunneling or just shorthand for unexplained reaction rate enhancements.
And that's where much confusion arises. If we label every unexplained phenomenon quantum, we drift into speculation. Biologists want conclusive evidence, direct measurements or manipulations of tunneling states, not just rate anomalies that could be interpreted in multiple ways.
Fair, but the few rate anomalies that are tough to explain might indicate that tunneling is real, even if it's limited to special circumstances. The onus is on experimentalists to develop tools that isolate these effects in natural, living conditions.
Could quantum tunneling also cause DNA mutations through proton shifts?
Possibly, but most suspect its impact is minimal. Oxidative damage and replication errors already explain much of the mutation burden.
Still, the idea is captivating. Protons in the hydrogen bonds between base pairs might momentarily hop to an alternative site via tunneling, altering the standard pairing rules and leading to a mismatch. If replication happens before the proton reverts to its original position, a mutation gets locked in. Even if such proton tunneling events are rare, the vast number of cell divisions over an organism's lifetime could mean these events accumulate. The flip side is that known mutagens and replication slips occur far more frequently, so it's difficult to tease out if tunneling is adding a significant fraction to the overall mutation rate. Another complexity is that cells have multiple repair mechanisms. DNA mismatch repair and proofreading polymerases might catch these transient errors. That means any tunneling-induced mistake must slip past corrective processes. It's hard to confirm whether a mismatch is due to a quantum proton shift or just a routine replication slip, especially in vivo.
Right, the fundamental challenge is distinguishing quantum-driven errors from a sea of alternative explanations. Until there's an experiment that unequivocally shows a proton hopped via tunneling and not a classical vibration or thermal fluctuation, many will remain unconvinced.
Why does this discussion create such tension between physicists and biologists?
Physicists are trained to see quantum effects as the bedrock of reality. From the small-scale perspective, everything is inherently quantum. But many biologists are pragmatic. They find that classical or semi-classical explanations work extremely well for most experimental data. In complex systems, there's often no obvious reason to assume quantum tunneling is the key to explaining what we observe. And additionally, physicists might be more eager to push the frontiers, suggesting that nature could exploit quantum mechanics in ways we haven't fully recognized. Biologists, on the other hand, emphasize the complexity and noise of living systems. They ask, why would evolution rely on fragile quantum states, given how easily they decohere under warm, wet conditions? The tension isn't just about data, it's also about how each discipline frames research questions and what it considers strong proof.
Precisely. There's also a cultural divide. In physics, the idea of quantum weirdness is embraced. Physicists look for it everywhere. Biologists are often more interested in data-driven findings that link to known pathways and robust, reproducible measurements. So if a claim doesn't align with established biochemical models, the bar for proof is correspondingly high.
And that is exactly why you'll hear energetic arguments from both sides. Some see quantum tunneling in biology as the next frontier, while others view it as a distracting speculation unless we see more direct experimental evidence.
Can breakthroughs in nanotechnology help us finally test tunneling in biological systems?
Likely, yes. Engineers are already building single-molecule devices that measure tunneling currents. Applying those designs to, say, an enzyme or a DNA strand could reveal whether quantum tunneling steps occur in real time.
One promising approach is creating nanogaps that function as tiny sensors. When a molecule bridges the gap or alters its local environment, the tunneling current changes measurably. If these sensors are integrated with microfluidic systems, researchers could watch in detail as enzymes catalyze reactions or as DNA undergoes replication. They might detect signatures that match tunneling theory, for instance. Sudden current changes not explainable by thermal noise alone. Moreover, new computational tools could simulate these events with quantum-level detail. If the simulations predict a strong tunneling contribution and the experiments match those predictions, that will provide more confidence. But this path isn't easy. Designing experiments where a single enzyme or DNA strand remains stable for extended observation is challenging. Plus, the noise in such minute measurements can be enormous. Yet, the prospect of combining advanced nanotech sensors with molecular biology is exciting. It may yield a final verdict on whether tunneling is just a curiosity or a genuine driver of certain biological functions.
Precisely. Another possibility is manipulating enzymes or DNA to create conditions that favor or hinder tunneling. For example, strategic mutations in an enzyme's active site might either enlarge or shrink the distance for proton transfer. If reaction rates dramatically shift in line with what tunneling models predict, it would be a strong indicator that tunneling plays a direct role.
Traditional enzologists often say, we've explained enzyme activity thoroughly without quantum tunneling. Isn't this just an attempt to sensationalize the field?
I see that viewpoint, but I'd argue there are still puzzles. Some enzymes exhibit rate enhancements or temperature dependencies that aren't trivial. Maybe classical models can be refined to fit these data, but we shouldn't dismiss quantum explanations out of hand. There's a possibility we're missing a deeper mechanism.
And from the vantage point of DNA mutations, we face the same clash. We can account for most errors without needing tunneling, but the fact that a small fraction remains unaccounted for could open the door to quantum phenomena. Even if tunneling is a minor factor, it might still have evolutionary repercussions.
Indeed, small effects can become significant over large timescales or numerous replication cycles. That's a subtle point. Evolution is all about the accumulation of countless events. If tunneling adds even a tiny fraction to the overall mutation landscape, it might shape outcomes in unexpected ways.
To wrap up, let's summarize the core positions. In nanotechnology, there's zero dispute about tunneling. It's real, measurable, and already powering advanced electronics. The discussions there focus on how best to optimize or mitigate tunneling.
Right. In biology, the contention is significant. Skeptics see classical explanations as sufficient. Advocates point to unusual reaction rates and isotope effects as evidence that tunneling can't be ignored.
Could future research decisively settle this?
Potentially, if we manage to directly observe tunneling events in live cells, or even in carefully controlled in vitro systems, it would change the conversation. Until then, many will remain cautious.
Collaboration between physicists, chemists, biologists, and engineers is crucial. Quantum tunneling is a delicate phenomenon to observe, especially in a cellular environment where noise is high. But the rewards for proving or disproving its biological relevance are considerable. On one hand, we might discover new strategies for drug design or energy use in cells. On the other hand, if we find tunneling negligible, we can redirect resources to other avenues. In the meantime, the speculation itself drives methodological innovation, new detection methods, advanced computational models, and interdisciplinary partnerships. Whether tunneling in biology turns out to be a major factor or a minor curiosity, the pursuit of answers expands our scientific toolkit.
Exactly. The quest is as important as the final verdict. If nothing else, it pushes us to refine our understanding of both quantum physics and molecular biology. And From that effort, new technologies, especially in sensing and imaging, are already emerging.
On that note, we'll close this extended edition of Quantum Pathways. We've seen how quantum tunneling is a cornerstone of modern nanotechnology, even as its role in biology remains a fascinating puzzle. We hope our combined short Q&As, heated debates, and in-depth discussions give a richer view of the science, the controversies, and the future directions.
Thanks for tuning in. Stay curious, keep questioning assumptions, and remember that scientific disagreements are often where real breakthroughs happen. Whether you're a biologist, physicist, or just intrigued by the quirks of the quantum world, we invite you to keep exploring.